Hello. In today's video, we shall learn in detail about genus Pinus. By the end of this video, you will have a clear understanding about the internal structure of Pinus stem, pine needle, secondary growth, reproduction in Pinus with detailed account of male and female cones, male and female gametophytes, pollination, fertilization, and development of Pinus embryo. Let us start with the introduction to Pinus. Pinus is the largest genus of family Pinaceae under order Coniferales. Pinus are tall, erect, coniferous, evergreen, resinous, perennial trees popularly known as pines. Pines are gymnosperms or naked seeded plants in which the seeds are not enclosed within the fruit or ovary. They are most widely distributed in the northern hemisphere. Now, let us look into the morphology of Pinus. Pine is a tall, erect, evergreen tree. It has a taproot with well-developed lateral roots. The roots of Pinus have a symbiotic relationship with nitrogen-fixing bacteria. Young roots are infested with a fungus to form mycorrhiza. There are numerous aerial branches with green, needle-like foliage leaves. The stem is cylindrical, erect, rugged, and covered with scale bark. Branching is monopodial. The branches grow spirally and are of two kinds, long shoots and dwarf shoots. The long shoots have an apical bud which grows indefinitely and therefore show unlimited growth. Scaly leaves are present which are protective in function. The dwarf shoots, which are devoid of apical bud, have limited growth. They arise on the long shoot in the axil of the scaly leaves. Leaves are of two kinds, long, green, needle-like foliage leaves borne only on dwarf branches, and small, brown, scaly leaves borne on both kinds of branches. A dwarf shoot with its foliage leaves is called spur. The sporophyte plant body is differentiated into root, stem, and acicular or needle-like leaves. Cones, which are seed-bearing structures, are present in Pinus. Pinus is monoecious. Male and female cones are seen on separate branches of same plant. Female cones are larger than the male cones. Seeds are mostly small and winged and are anemophilous. Next, let us study the internal structure of the young stem of Pinus. It shows an irregular outline of epidermis. In Pinus, it is the outermost single layer of cells with a thick cuticle. Just below the epidermis is a layer of lignified cells that form the hypodermis. Hypodermis is followed by many layers of more or less rounded, thin-walled parenchyma cells forming the cortex. Many conspicuous resin canals or ducts lie embedded in the cortex. These resin canals are surrounded by a layer of resin-secreting glandular epithelial cells. Internal to the cortex is a single-layer, thin-walled endodermis, followed by pericycle, made up of parenchyma cells, but sclerenchyma cells are absent. Vascular bundles are conjoined, collateral, and open, arranged in the form of a ring. Each bundle is made up of outer phloem, middle cambium, and inner xylem. Phloem consists of sieve tubes and phloem parenchyma. Xylem consists of annular and spiled tracheids with no true vessels. Protoxylem is formed towards the center, while metaxylem towards the periphery. Medullary rays are seen between the vascular bundles. There is a well-developed pith in the center, and it consists of mass of parenchyma cells. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next, let us understand about the secondary growth in pinus. Secondary growth takes place when the cambium becomes active and cuts off secondary wood or xylem on the inner side and secondary phloem on the outer side. The secondary wood consists of tracheids with bordered pits on their radial walls. Distinct annual rings are seen consisting of the autumn wood, which are made up of narrow, thick-walled tracheids, and the spring wood made up of wider and thin-walled tracheids. Such a compact wood is called pycnoxylic. There are numerous horizontal medullary rays. The xylem portion of the ray consists of thick-walled ray parenchyma and short tracheidal cells. The phloem portion of the ray consists of large, thin-walled cells called albuminous cells. The cork cambium cuts cork and bark towards the outer side and some parenchyma or secondary cortex towards the inner side. Many resin canals and stone cells are present in the secondary cortex. Central part of the stem is filled with parenchymatous pith. Internal structure of the pine needle. It shows the presence of single layer of thick-walled cells of epidermis covered by a strong cuticle. It is interrupted by many sunken stomata, which opens internally into a respiratory cavity or vestibule. Below the epidermis lies a few layers of thick-walled sclerenchyma cells forming the hypodermis. It is interrupted by substomatal cavities. Just below the hypodermis, resin ducts are present. Each resin duct is made up of layer of secreting epithelial cells which are surrounded by a sclerenchymatous sheath. In between the hypodermis and endodermis are seen a mass of large, thin-walled parenchyma cells. These are the mesophyll cells which contain chloroplast and starch grains. They show peg-like infoldings of the wall. These infoldings or projections increase the photosynthetic area of the cells. Internal to the mesophyll layer is a single layered endodermis formed by barrel-shaped cells with Casparian strips on their radial wall. Next is the pericycle, which is a multi-layered part. It consists of parenchymatous tissue densely filled with starch grains, albuminous cells near the phloem filled with proteins and starch grains, and tracheidal cells near the xylem with bordered pits. Albuminous cells and tracheidal cells are together known as transfusion tissue. They help in the lateral flow of nutrients. Vascular bundles, generally 2 in number, lie embedded in the multi-layered pericycle. They are collateral and closed. Xylem lies towards the angular side of the leaf and the phloem towards the convex side of the needle. Next, let us study about reproduction in Pinus. Pinus plant body is a sporophyte. Pinus are gymnosperms that produce seeds in cones. The plant is monoecious but bears both male and female cones on separate branches of the same plant. Male cones develop on lower branches while female cones appear on the upper branches. The male cone consists of microsporophylls and the female cone consists of megasporophylls. The male cone is a small oval-shaped structure which consists of number of microsporophylls arranged spirally round the axis. Each microsporophyll has a stalk, a terminal leafy expansion, and a tip bent upwards. It bears 2 pouch-like microsporangia or pollen sacs on its under surface. A mature microsporangium consists of a multi-layered wall, tapetum, and microspore mother cell. Each microspore mother cell undergoes reduction division to produce a tetrad of haploid microspores or pollen grains. Each pollen grain is covered by an outer exine and an inner intine. The exine forms the wings, one on each side. On elongation of male cone, microsporangia are exposed and dehisce longitudinally, dispersing the pollen. The female cones are generally produced singly or in pairs, spirally in the axil of the scale leaves on long shoots. Each female cone has an axis. Many megasporophylls are arranged spirally on the cone axis. Each megasporophyll consists of 2 types of scales, namely the Bragg scales and the ovuliferous scale. The Bragg scale or the carpellary scales are dry, small, thin, brownish scales. The ovuliferous scale is present on the upper surface of each Bragg scale. They are woody, stouter, bigger, and triangular in shape. A broad sterile structure with pointed tip is present at the apex of these scales. Each megasporophyle bears 2 ovules on the upper surface. Each ovule is orthotropous and consists of a central mass of tissue, the nucellus or megasporangium. It is surrounded by a single integument consisting of an outer fleshy, a middle stony, and an inner fleshy layer. An opening or gap in the integument forms the micropyle. Just opposite to the micropyle is present a pollen chamber. 2 to 5 archegonia are present in the female gametophyte or endosperm. Within the nucellus, a megaspore mother cell undergoes meiosis to produce 4 megaspores in a linear tetrad. 1 megaspore remains functional while the other 3 degenerate. Let us now look into the male gametophyte. The microspore or pollen grain divides to form 2 prothallal cells and an antheridial cell. The antheridial cell divides and forms a generative cell and a large tube cell. The prothallal cells become disorganized even prior to pollination. The pollen grain sheds at this stage and further changes take place after pollination. Nearly a complete year is required for the entire development of the male gametophyte. Next is the female gametophyte. The megaspore germinates in situ or within the ovule and forms the female prothallus or female gametophyte within the nucellus. It undergoes free nuclear division, forming a solid mass of tissue called the endosperm. It surrounds the embryo in the seed. At the micropylar end of the prothallus develop 2 to 5 archegoniae. Each archegonium consists of a solen venter and a short neck. The venter comprises of a large egg cell with a distinct egg nucleus and a ventral canal cell. The archegonial wall is made up of small jacket cells. The archegonia mature slowly and in the following year become ready for fertilization. Next, let us study the development of male gametophyte before pollination. The pollen grains undergo endosporic development. The pollen nucleus divides mitotically to produce a small primary prothallal cell towards the proximal end and a large central cell towards the distal end. The central cell divides to form the secondary prothallal cell and an antheridial initial. The antheridial initial divides to form a small generative cell and a large tube cell. The pollen grain is now showing a 4-cell stage, that is, 2 prothallal cells, a generative cell and a tube cell. Pollination takes place at this stage. Next is the development of male gametophyte after pollination. After pollination, the 4-celled pollen takes about a year to germinate. In the next spring, the pollen forms a pollen tube and comes out through the aperture. It moves towards the archegonium and penetrates the nucellar tissue of the ovule. The generative cell or the antheridial cell within the pollen tube divides to form a stalk cell and a body cell, otherwise known as spermatogenous cell. The body cell divides and forms two male nuclei or gametes just prior to fertilization. Next, let us study the development of female gametophyte. The female gametophyte develops from the functional megaspore. A large number of nuclei are formed by the mitotic divisions of the nucleus of megaspore. With the increase in size, the megaspore develops a vacuole in the center, and the nuclei are found around the vacuole embedded in the cytoplasm. The gametophyte becomes cellular by wall formation and represents the endosperm or the female prothallus. Few cells of the female gametophyte at the micropylar end enlarges, has dense cytoplasm with prominent nuclei. These cells function as archegonial initials. The archegonial initials divide to form the neck initials and the central cell. The neck initials form the neck cells. The central cell divides to form the ventral canal cell and a large egg cell. A nutritive layer called archegonial jacket, made up of small cells, surrounds the archegonium. Pollination is anemophilous. Pollen grains are dispersed by wind. The mucilate secreted at the base of the micropyle traps the pollen grains and draws them into the nucellus of the ovule. After pollination, the mouth of the micropyle gets closed. Fertilization takes place in the following year when the archegonia mature. The exine of the pollen grain bursts and the intine forms a slender pollen tube. It enters the tip of the archegonium. The tube nucleus moves into the pollen tube, divides and forms a stalk cell and a body cell. The body cell divides and forms two non-motile male gametes. The pollen tube bursts and releases the two male gametes. One of the male gamete moves to the egg nucleus and fuses with it to form the oospore. The other male gamete, the stalk cell and the tube nucleus disintegrate. Finally, we shall look into the development of the embryo. The oospore undergoes two successive mitotic divisions within the egg cell and forms 4 nuclei. Another division results in 8 nuclei arranged in different tiers. The lower group forms the embryonic tier. The embryonal cells divide to form embryo. Next tier is the suspension tier, which elongates to form embryonal suspension. It pushes the embryo into the female gametophytic tissue. The other tiers are nutritive tiers, which provide nourishment to the growing embryo. As there are a few archegonia in the ovule, some more embryos may be formed, known as polyembryony, which is a very unique characteristic of pines and conifers. Only a single, strong, deep-seated pro-embryo develops into an embryo while the rest degenerate. The embryo consists of an axis with a radicle, a hypocotyl, a tiny plumule with a number of cotyledons. After fertilization, the ovules develop into seeds. The seed is provided with a well-developed, thin, papery, membranous wing, which is formed from the outer fleshy layer of integument and a part of ovuliferous scale. Germination of seed is epigele. So today we have studied in detail about the internal structure of pinus stem, pine needle, secondary growth in pinus stem, reproduction in pinus with a detailed account of development of male and female gametophyte, pollination, fertilization, and finally development of pinus embryo. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.